Alrighty, so we've reached the last chapter of the course for AP1, special senses. So let's talk a little bit about the special senses. So what exactly are special senses? Special senses are those that convey very specific stimuli from very specialized sensory organs to discrete locations within the head, the brain. These things are things like smell, which we refer to as olfaction, taste, which is gustation, vision, hearing, and hearing is called audition, and vestibular sensation. The special and general senses differ in some very significant ways. One way is through the stimuli detected. The general senses are those senses that involve touch, pain, and temperature. Think about things you can do with your hands. The special senses has special neurons which detect very specific stimuli, things like light and sound, the movement of your head, and chemicals. It's when we taste and smell things, what we are smelling and tasting are chemicals. Uh, Structures of the sensory receptors are very different. In the special senses, there are receptor cells. They're not neurons. The general sensory receptors are the receptive ends of sensory neurons. So the things that detect when we touch something, that's the end of a neuron. But when we smell something, that is a specific cell within that organ that is doing the sensing. Finally, the location of the sensory nerves. Sensory nerves are found in organs, all confined to the head. All of that information travels on axons of very specific cranial nerves, and general sensory information travels on both the axons of both cranial and spinal nerves. So, special senses are those things found within the head. And there is a process called transduction. Did I skip a slide? Okay. Uh, Transduction is the physical or chemical stimulus that is converted into an action potential that can then be interpreted by the brain. The neurons involved in general senses have special receptive ends. They detect touch, temperature, and pain. These stimuli alter resting membrane potential of the sensory neurons to produce their action potential, and the add-action potential then reaches the central nervous system. The special senses also detect stimuli, and again, light, sounds, chemicals, and transduce them into action potentials. These are propagated through axons of peripheral neurons to the central nervous system. So the actions of sensory transduction is different. Stimuli for both general and special senses are first processed by sensory nuclei and transmitted to the thalamus and to the primary cortex areas for awareness and identification. Sensory signals would then pass to association areas for further interpretation and integration. The first stimuli received is you hear something. It's a sound, it's off in the distance, it's a high-pitched wail. That signal is then transmitted to the thalamus, then to the primary cortex, where you recognize it as a high-pitched wailing sound. It then goes to other cortex areas where you become aware, yes, that is a siren, and it is coming toward me. And if you've been around long enough, you know whether that siren is a fire truck, an ambulance, or a police car. And there's the figure 15.1, which is the comparison of general and special senses, showing the stimuli being detected, touch, pain, temperature, with general senses. There's the receptive end of that monoaxonic neuron. See the single, there's the cell body, single axon and dendrite, and there's the receptive ends. Special senses, taste, light, sound, head movement, and smell. You see the smell is a little bit different, but for taste, light, sound, and head movement, you have these special sensory cells, which are attached to the axon ends, the sensory neurons. The smell is slightly different, where you have receptive endings on the olfactory neurons. So, smell. Human beings have a good sense of smell, but it is not anywhere near what some other organisms are. And you will see in a moment, based simply on the number of slides that you're going to see and the time we're going to spend talking about that sense, which sense is most important for human beings. So, the structures of olfaction. The olfactory system allows for the detections of odorants. Those are things we would consider smells. Therefore, if you don't want odorants to emit from your body, what do you put on your body? You put a deodorant on. So smell signals in the air are referred to as odorants, transduces them into signals perceived as odors. Olfaction is initiated in the olfactory epithelium, which is a small region of specialized cells in the superior nasal cavity. I'm sure you've been through this in lab and you've looked at that specialized epithelium found in the nasal cavity. The olfactory neurons are modified bipolar neurons. So if we'll back up a couple of slides, see, bipolar neuron, unipolar neuron, bipolar neuron, but you see the specialized cells at the end for the chemoreceptors. And those chemoreceptors detect chemical substances which our brain then perceives as odors. Basal cells, which are stem cells, they continuously replace olfactory neurons. These cells have a lifespan of 30 to 60 days. One of the reasons our sense of smell deteriorates as we age is our ability to replace these cells slows down as we age. And then finally, there are supportive cells, which is a group of columnar cells which surround the olfactory neurons and basically hold them in place. The olfactory neurons are combined axons from the olfactory neurons form the olfactory nerve, cranial nerve number 1. The olfactory bulb sits above the cribriform plate in the ethmoid bone, inferior to the frontal lobe of the brain. And when you look at a model of a brain and you turn it upside down, you see these two little structures that look like little light bulbs on the end of a little stalk. That is the olfactory bulb. The olfactory tract is the axon that exits the olfactory bulb, travels to other regions of the central nervous system where we get all of our interpretation done. If the olfactory bulb looked like the little light bulb when you turned it upside down on the brain, the olfactory tract is the little cord running from the light bulb. And again, there is the olfactory epithelium right there. This is the nasal cavity. Here, the inhaled air containing its odorants. They are then picked up by these specialized cells. Here's the columnar cells holding everything in place. There's the nerve cells, again, unipolar nerve going up, attaching to the olfactory bulb, which again, you can't see it very well on this particular drawing, but that there's sort of a bulb-looking structure right there. And then all of these nerve fibers come together to form the olfactory tract. Odorants in inhaled air are detected by the olfactory neurons. A chemical stimuli is transduced into an electrical signal, which is transmitted to various regions of the brain for identification. The olfactory receptors are activated when odorants are dissolved in the mucus surrounding the olfactory neurons' cilia. Then odorant binding proteins transport those odorants through the mucus to the receptors on the cilia of the olfactory neuron. As we're going to say a little bit later with taste, if something isn't soluble, if it doesn't dissolve, you don't smell it. The events that happen during transduction, first, the odorant is bound to a receptor and activates a G protein. The activated G protein then triggers the enzyme adenylate cyclase, to convert ATP into cyclic AMP. That cyclic AMP opens ion channels, which allows sodium and calcium ions to enter the cell, which causes a depolarization, which creates an action potential. And then the threshold, if that action potential threshold is reached, the odorant can be transduced from a chemical stimuli to an electrical signal, and that is when we smell it. So here is your olfactory cilia, the olfactory neuron. There's the ECF, the extracellular fluid. The odorant gets bound to the receptor, activates the G protein. The G protein moves over, produces the cyclic AMP. Cyclic AMP opens, allowing for calcium and sodium to pass through. The olfactory pathway is where the neural signals carry that information to various regions of the brain for detection and identification. Axons of the olfactory neurons carry those stimuli to the olfactory bulb in the central nervous system. The neurons, axon terminals, synapse. We remember what synapse means with the dendrites of mitral cells. These are the neurons found within the olfactory bulb. The mitral cells then form the bulk of the olfactory tract, and they travel between the olfactory bulb and the primary olfactory cortex, which is located in the temporal lobe of the brain. The olfactory tract is the only sensory pathway that bypasses the thalamus. Probably a good idea to remember that come test day. The olfactory tract is the only sensory pathway that does not include the thalamus. The primary olfactory cortex is responsible for awareness and identification of odors. Some other parts of the brain, the amygdala, the hippocampus, the hypothalamus, and components of the limbic system receive information from the primary olfactory cortex. These invoke both emotional and visceral responses to odors. You may smell a smell around the holidays, and it may make you think of your grandmother that you've recently lost. That would be an emotional response. You may smell something that reminds you of something very unpleasant. I don't know, maybe you were once in a really bad car wreck right next to a bakery, so every time you smell bread baking, you go back to those visceral responses of that automobile accident. Anosmia is a lack of olfaction. This is where you cannot smell things. This can be a very serious condition because you can't detect the smell of smoke. You can't detect the odor of spoiled food. People may suffer from malnutrition. Olfaction contributes significantly to our sense of taste, so food loses its appeal. Some people will tell you that the sense of taste is about 40% smell. If things don't smell right, they don't taste right. I mean, how many of us have had a cold where, really bad cold, your head's completely clogged up, you can't hardly breathe, and food just doesn't taste the same, and you lose all appeal. The appeal of eating goes away. Many conditions result in anosmia and hyposmia, which is a reduced sensitivity to smells. Anything that blocks air from reaching the olfactory epithelium will interfere with olfaction. Nasal polyps, a deviated septum, a fractured nasal septum, swollen respiratory epithelium, or allergic reactions. Many of these situations are in fact temporary, and once airflow is restored, the olfaction returns. However, anosmia can be permanent. Head injuries that damage neural pathways at any point along the path could disrupt olfaction. The olfactory nerve is particularly vulnerable for fractures to the cribriform plate. Cribriform plate is found at the upper end of the nose. I'm pointing to my nose, you can't see me. But boxers, for instance, MMA fighters, will many times have a very decreased sense of smell because of the repeated punches they've taken to their nose. Neurodegeneration of Alzheimer's and Parkinson's patients often impairs the sense of smell. Hyposmia may be an early sign of Alzheimer's disease. Lots of research being done in Alzheimer's, and we are finding that lots of those patients, one of the first things they started to notice, or their family records that they started to notice, was their sense of smell wasn't what it once was. It can also develop naturally with aging. Again, as there's fewer olfactory neurons are replaced by basal cells. This occurs gradually, but especially noticeable after the age of 60. I will be turning 60 next year, so that's something I'm going to be looking at right now. My sense of smell actually isn't what it should have been. When I was in graduate school, I did some work in the Fish Museum at UALR, and through some no fault of the school, this was totally my fault, I damaged my my sense of smell with a product called formaldehyde. Didn't wear the proper mask. All right, we're going to stop here and we will talk about taste at another time.